In the late 1940s, researchers isolated a new class of antibiotics from the soil bacterium Paenibacillus polymixa. One of these compounds became known as colistin. Early clinical use in the 1960s revealed severe side effects. Colistin caused dose-dependent damage to the kidneys and the central nervous system. Once safer, better-tolerated antibiotics were developed, it was largely abandoned. By the early 2000s, intensive care units faced a crisis that forced them to pull this highly toxic drug back to the front lines. Gram-negative pathogens, specifically carbapenem-resistant Enterobacterales and Acinetobacter baumannii, were multiplying unchecked, having developed resistance to nearly every modern drug class. Colistin was one of the very few agents left that could still kill them. This reliance comes with immense risk. Colistin has a narrow therapeutic index. The margin between a plasma concentration that clears the infection and a concentration that induces acute kidney injury is razor thin. To deploy this tool safely, critical care teams have to understand the precise physical mechanics of how the molecule operates and exactly how bacteria are evolving to survive it. Colistin targets the outer membrane of gram-negative bacteria, specifically the negatively charged lipid A molecule. Bacteria use positively charged calcium and magnesium ions to stabilize these repelling charges. However, colistin's overwhelming positive charge forcefully displaces these stabilizing ions, binding directly to the lipid A. This creates a detergent-like effect. The outer membrane loses its structural integrity, violently rupturing and leaking the cell's internal cytoplasm. Because of its severe toxicity, intravenous colistin cannot be administered in its active form. Instead, it is given as an inactive prodrug called colistomethate sodium, or CMS. CMS hydrolyzes slowly and spontaneously in the bloodstream, eventually releasing the active colistin base. This slow in vivo conversion requires doctors to administer a massive initial loading dose of exactly 9 million units, or about 5 milligrams per kilogram of colistin base activity, to quickly reach therapeutic plasma concentrations. This loading dose is mandatory regardless of a patient's kidney function. Failing to hit the therapeutic threshold immediately is directly linked to early treatment failure in critically ill patients. Maintenance doses are only adjusted for renal clearance after this initial strike. Even with precise dosing, colistin exhibits heteroresistance. Minor bacterial subpopulations can survive standard drug concentrations. To suppress these survivors, colistin is almost never used as monotherapy, but rather paired with agents like carbapenems or tigecycline. Colistin's reliance on electrostatic physical attraction makes it highly effective against standard membranes, but it also creates a clear mechanical pathway for bacteria to evolve resistance. Certain genera, like Proteus and Serratia, possess intrinsic resistance to colistin through natural, constitutive modifications to their outer membranes. For normally susceptible species, acquired resistance emerges through spontaneous chromosomal mutations. These typically occur in two-component regulatory systems, involving genes like MGRB, PMRAB, and FOPQ. These mutations command the bacteria to synthesize and attach molecules like ARA4N and P-N directly to the lipid A. This addition reduces the net negative charge of the outer membrane, neutralizing the electrostatic attraction and successfully repelling the colistin molecule. In 2015, a second highly mobile pathway was identified in Escherichia coli, isolated from food animals in China, the MCR1 gene. Unlike chromosomal mutations, MCR genes reside on conjugative plasmids. This allows the resistant sequence to be horizontally transferred, spreading rapidly across different species of bacteria. Because these plasmids frequently co-carry other resistance determinants, their agricultural and environmental dissemination has triggered a massive infection control crisis across human, animal, and food domains. Chromosomal mutations emerge under direct antibiotic pressure within a single patient. Plasmid-mediated MCR genes create an infectious epidemiology of resistance, turning colistin failure into a transmissible threat. To counter these resistance mechanisms, clinical microbiologists must first detect them. But for colistin, standard susceptibility testing procedures reliably fail. Agar disk diffusion is the routine method for profiling most antibiotics. Both American and European clinical guidelines explicitly forbid using it for colistin. Colistin is a large, amphiphatic, and polycationic molecule. Instead of migrating evenly through the agar matrix, it ends up binding aggressively to plastic labware. Because the drug fails to reach the bacteria in the dish, isolates that are actually resistant will appear susceptible. This false susceptible reading can easily mislead a physician into prescribing an ineffective treatment. The only internationally endorsed reference method for colistin is broth microdilution, performed in cation-adjusted Mueller-Hinton broth. Under current guidelines, an isolate is classified as susceptible only if its minimum inhibitory concentration, or MIC, is less than or equal to 2 mg per liter. An MIC greater than 2 mg per liter confirms colistin resistance. The practical flaw with this gold standard is time. Broth microdilution is highly labor-intensive and requires 16 to 20 hours of strict incubation. For a critically ill patient in septic shock, waiting 20 hours for a manual laboratory result often guarantees the treatment arrives too late. To bridge the gap between diagnostic accuracy and urgency, laboratories are turning to CRISPR-based diagnostics. These platforms rely on DNA or RNA targeting nucleases, primarily Cas12a or Cas13a, paired with an initial isothermal amplification step, like recombinase polymerase amplification. Because this amplification is isothermal, it operates at a constant temperature. This eliminates the need for expensive thermal cyclers, requiring only a basic heating block. In the assay, a precisely engineered guide RNA searches the extracted sample to perfectly match and bind to a specific MCR sequence, activating the Cas nuclease. Once activated, the enzyme indiscriminately cuts nearby reporter probes, releasing a highly visible fluorescent signal. The entire workflow, from rapid nucleic acid extraction to a visual readout, takes approximately 60 minutes. This speed and portability enable decentralized surveillance. Critical resistance data can be gathered directly in hospital wards, primary care settings, and agricultural sites in low-resource settings. However, CRISPR diagnostics have a strict limitation. They only detect the specific MCR sequences their guide RNAs are programmed to find. They are entirely blind to the spontaneous chromosomal mutations in genes like MGRB, which remain the most common resistance mechanisms globally. A positive CRISPR result instantly confirms a transmissible MCR gene, but a negative test still requires traditional phenotypic testing to rule out other forms of resistance. We are now forced to deploy modern single nucleotide molecular screening just to safely administer a highly toxic cyclic peptide discovered in 1947. Understanding exactly how this molecule binds and how bacteria evade it is the only way to preserve its utility in the intensive care unit.